So our case, I'm very happy for Lina that she was introducing to us GBIF, because we are actually working with GBIF data. Because our aim is to actually integrate this occurrence data with satellite data and machine learning techniques, because we want to investigate how good are these data in habitat and species distribution modeling, especially for citizen science and museum data, which are not really included that often, at least in my experience. It's more research data that are chosen for this type of modeling. And also, so we want to inspire also museums, collections, or also research groups to share more of their data sets in GBIF. Because I saw that lots of groups are really keeping the data for themselves. And also, we want to refine the habitat classification system over Europe, which are mainly based on, at the beginning, on knowledge, personal knowledge of experts, and then the probability maps that were created after that are again based only on research data, but are lacking huge data set, I will show you. And so what is actually this system? This habitat classification system it was implemented by the EA, and it's a hierarchical system that gives a framework for classifying the habitat all over Europe of natural and natural habitats. And this is based, as I said, from at the beginning, it was an expert group that was classifying these based on physical factors, the dominant vegetation types, not the real occurrences of species, but just groups of species or percentage of coverage, for example, or also using just ecological processes and human influence, and based also on current land use. So it is good, but it is not really, at least in our experience, not really precise. And so they created this system of classification, which started from a simple sentence, for example, how much water is present, and then, or it is like 70% or less, and then you go on like yes, no, and to the next question until you get the class of the habitat. And then many years later, they also produced probability maps, which are based on vegetation plots from the European Vegetation Database and Brown Blanket Database, which again only research data sets. And so what we actually saw when we had a look on GBIF data and compared to these probability maps is, as you can see, in red you have the study case of our group, which is the habitat S22. And many times, for example, for the species Lepidodendron ferruginum, which is one of the main species of the habitat, this was falling outside. So we wanted to investigate, what about we use the real occurrences, especially from GBIF, to see if this can be a bit more accurate. And why also GBIF? Because it contains so many data that are not that used. For example, citizen science project, 15,000, or museums, not so many, but still data that are not present in the European vegetation plots database. And again, plotting the data, as you can see in green, is again citizen science data, while in blue, only in blue, the research data. And museum, there is something in red around Europe, but there is still so much potential and people are not sharing this data. So our workflow was starting from choosing one habitat from the EUNIS classification as S22. Then we were taking the main domestic species, which are used as indicator of such habitat. And then we were retrieving the GBIF data occurrences. We were also choosing some environmental predictors from environmental satellite data, and then combining them in an occurrence environmental dataset. And And then we were also generating pseudo-absence data because we were only working with presence data from GBIF. So then with the complete data set, we ran individual models, so GAM, GLM, and RF, random forest, produce also TSS scores, and which were the base to choose the best predictions from the sample model, which then gave us the prediction maps for each species, which we also validate with the EVA, so this vegetation plot database, to see really, okay, is there does our data make sense with the research data, let's say. And then, and yes, but I will see. And then we derived an aggregated habitat from all these prediction maps. And we compare lastly with the EUNIS habitat probability map. And so at the beginning, what we saw through a regression test of our data and the environmental predictors is that actually our the earliest definition of the habitat was making sense with what we found, how the species were found in the ecological context, because we saw that indeed they are found in mountain and alpine areas, and they need stable, not excessive water availability. And as the EUNIS habitat said, that it depends on the dominant species where you find this habitat, because I will show you later. There's one species, especially this Vaccinium ingenosum, that is a bit divergent from all the other main species of this habitat. In fact, it's more focused on Scandinavian regions, which are ecological a bit different from the Alps and all over these other mountain European areas. And so what we had were these 8 main species and with prediction maps for them. In black, you can see the pseudo-absence made predicted from the presence occurrence data. And then we had in blue the predicted distribution ranges. And in the end, this point of the workflow, we are working on comparing our results with EUNIS probability maps. And here, so we are working on 2 resolutions, one at 1 kilometers all over Europe, and another only for the Alps at 100-meter resolution. And when we check all over Europe, so in particular, the red areas is the EUNIS ranges, while in blue it's our results. And when we were using all 8 species, we could only see that they were overlapping, or there were some differences in over Europe, but not Scandinavia. So then we reduced the number of species we used, and again, finally we had our ranges also in Scandinavia, which actually agrees with the definition of the EUNIS habitat, because they were saying you can have this habitat more or less in mountain regions of Europe, but it depends on the dominant species. So when you have Vaccinium or some others, more typical Scandinavia, then you can have this habitat also in that area. And while instead we compare the Europe, like the Alp area, which is a bit easier to see our results, the comparison. So we can see that in the blue areas are a bit more higher elevations than a broader and also lower elevation EUNIS distribution for the habitat, while in purple is the overlap. So overall, the overlap seems to be enough, but also we can see some differences. We are still working on comparison of all these results, but for now we can see that it makes sense also you use GBIF data as they are. And finally, we are sharing our results, our documentation and workflow in the many channels that very few projects provided us. And yeah, we are refining all the documentation in this period. Thank you for your attention.